and good morning to Dr. Farah. So today I would like to present on Stigmasterol as anti-inflammatory and antioxidant compound against cardiovascular disease. So based on my reading, there are 12 basic important steps in potent drug discovery and development. So step 1, I would like to further clarify on how I actually choose the disease type. So I chose CVD generally because it was disease which affect the first world countries. So I had come across the statistic on the causes of death in Malaysia at 2017, in which the percentage of death by ischemic heart disease was the highest. So step 2, we proceed with the identification of the drug target. So before I decided, I went through a journal which was published at 2010. In this journal, Gabe et al. Stated that the Stigmasterol able to bind to the chondrocyte membrane. So it is clear that chondrocyte membrane will be my target protein. Step 3, we move to the identification of hit. So at this stage, I will test on the compound against the target protein. So compound that will show activity will be pronounced as hit. It will be promoted as the hit. So in this case, some of the hits discovered from the phytochemical constituent of Alpha-nitrosaccharase are alpha-spinosterol, camptosterol, beta-carotene, and also stigmasterol. Step 4, I will further evaluate on the hits to lead compound. So the hits will be further examined to find the promising lead compound. So in this case, one promising lead compound series was discovered, which was the Stigmasterol. So in step 5, we will proceed with the optimization of the lead compound. So in this step, it is important because we want to assess for the toxicity, potency, the binding effect, and also the strength of the promising lead compound. So for the lead optimization step, there will be 5 important steps that I will do. The first one will be the synthesis of the analog for the promising lead compound. So how I will synthesize the analog is by identifying the structure-activity relationship between the compound and also the target protein. Next, identify the pharmacophore through the synthetic modification, conduct further experiment on different bioassay in which it will be either in vivo, in vitro, or ex vivo. Then determine the toxicity and also the efficacy in animal model, and then finally will be the pharmacodynamic and pharmacokinetic. I need to determine the pharmacodynamic and pharmacokinetic. So step 6, I will apply for the patent protection. Step 7, the preclinical trial will be conducted. And step 8, the IND application, I will apply for the IND. IND stands for the investigational new drug. For step 9, will be the phase 1. It will be the phase 1 in which in this phase, the drug will be tested on healthy volunteers in order to determine for the toxicity and to screen for the unexpected side effect. And then in the step 10, it will be the phase 2, in which in this phase, the drug will be tested on a small community of patients, in which I want to determine for any of the beneficial effect and also the dose level that need to be reached. And then at step 11, this one will be the phase 3. So in this phase, drug will be tested on larger group of patients, and I will compare with the existing treatment. And then in step 12 will be the new drug application. In this moment, I need to prepare for the formal proposal for the FDA to approve for the approval of this drug. And then in step 12 will be the phase 4. At this level, the drug will be placed on the market, and then the patient will be monitored for any of the incoming side effects. So after explaining on the drug development, I will proceed with how I would like to specifically design the drug. So since I have precise biological target and its structure, so I will consider to use a technological method, for example, such as the computer-aided drug design. such as the in silico drug design or molecular modeling, and also combination chemistry. And I will also consider to use a visual model in order to determine the interaction between the compound and the target. Next, I will also focus on the best physical chemical properties that will be used in this drug design. First properties will be this drug will have both basic and acidic properties, or we also know it as amphoteric properties. And then this is important for the absorption and also for the permeability, for better absorption and permeability. And then it is also important for having correct lipophilicity, because if it is too low, it will have poor permeability. If it is too high, it will have poor solubility, so it should be optimum. So I will also consider to increase the ionization and the hydrogen bonding interaction, as it will linearly increase the efficacy of the drug solubility. And then proper 3D arrangement of functional group of the molecule also is very essential in order to develop a strong interaction with the target protein. And then rigidification by introducing the ring, in which it can also be used in this drug design in order to lock the molecule so that it won't rotate freely, and then so that we can reduce the side effect. And then the isosteric or the bioisosteric replacement, isosteric and bioisosteric replacement, in which this will be helpful to increase the bioavailability and pharmacokinetic. We also can use this to reduce the unwanted side effect, and so that this drug also will be capable to cross any of the barrier. So I think that's all from me. Thank you.